Exonate's a company that we started in 2014 here in Cambridge with IP from Nottingham, Bristol, and the University of New South Wales. We got Sunil as chairman before we even incorporated the company, because as everybody in this room knows, Sunil does not think like ordinary people, and we wanted to do it differently. So we didn't want to be the usual go to BCs, get into bed with the devil and all that sort of stuff. And I thought that Sunil would be very, very helpful on that, and he has been. So at the moment, we're looking for our Series A. We've gone a long way without a Series A using non-dilutive funding, but we now need some serious cash to take our lead product through phase 2. It lasts for 3 years, which is about how long the phase 2 is going to take. To date, we have completed a phase 1 study in patients. We call it 1B2A. We could not have got a better result out of that study. I'll tell you about it in a minute. We have been really good at conserving cash. So we've only taken just under 5 million in equity financing, which has got us from molecules within a university, dumped them, made some more, filed patents, did phase 1, and have an active IND. They're our molecules, composition of matter, all the important ones are now granted and have a shelf life through to 2044. So these are good new molecules. We did a collaboration with Janssen, which was superb and excellent. We learned a lot. They paid for most things and it went very well. They took a strategic different term from us. So we are in the process of getting absolutely everything back from them and we're taking it forward ourselves. What we do is splicing factor kinases. So we make small molecules that hit an SRPK1 kinase in order to change the isoforms that VEGF exists in. Management team, I'm the co-founder and CEO. Loic, who's at the back of the room, is our chief operating officer. We have the first-in-class SRPK1 inhibitors. We are making topical applications as an eye drop for the treatment of retinal diseases. So we have managed to make molecules that put on the front of the eye, they go around the side and into the back. Which when we started this company, everybody said you wouldn't be able to do that. It wasn't possible. But we all know they used to say you couldn't fly either. We have some really good investors. We got a Seeding Drug Discovery Award from Wellcome. We have O2 Ventures, Parkwalk. IP Group sort of inherited a few shares by default, but they're an investor. And basically another load of angels, both Cambridge angels, but also we have other angels from around the place. This is, for those of you who know ophthalmology, this is the best clinical advisory board you can get. We've got the Jocelyn and Harvard, we've got Melbourne, Sarah, we've got University, the Queen of University in Belfast, and we've got Johns Hopkins. We have a very lean way of doing things in that we have clinical development team, none of who are employees for us. We'll hire them in as jobbing experts with over 25 years expertise each in every area that we use, and it works well. So we're doing topical eye drops, we're inhibiting SRPK1. It's a very large market. If you get diabetes, you are at risk of getting eye disease, and about 50% of type 2 diabetics will suffer vision loss. It's an enormous market. We're all getting older, we're all getting fatter, and a lot of us are turning diabetic. And this is currently treated by anti-VEGF injections once a month into your eye, which a lot of people are not that keen on. 40% of those patients have an inadequate response. So that is about what you would expect with what we're doing. 30% of people who are offered an appointment for an injection don't go because they're frightened and scared of it. No, I don't really blame them. Those people who have early stage disease, there's a wait and watch at the minute. They are not treated, so just wait until you lose some vision, then we will stick a needle in your eye. So it's a big market. We're trying to target the early stage patients who are at the moment having wait and watch. This is our phase 1 study. We did it in Australia. This criteria for entry means they had mild disease. So these are the wait and watch population. We did that. We thought it was unethical to not offer treatment to people who would need it. And also we wanted people who were naive of any treatment, and we could find them in the UK and Australia. It was the middle of COVID. We chose Australia for various business reasons, but it turned out to be very lucky because Australia shut the borders and continued. Our results. These are the endpoints: primary safety and tolerability. It was a first in human study, secondary systemic PK, and then we wanted to see, did this drop get to the back of the eye, and if it got there, did it do something? We met all our primary endpoints. Nobody dropped out, no serious side effects, nobody had any tolerability issues. It was as good as comfort. The comfort levels were as good as normal eye drops for dry eye. Secondary endpoints, we got exactly what we wanted in PK, in that short systemic half-life, therefore very little systemic liability. Wanted to show you these. Right, top line of 5 patients at the screening visit. The yellow circles show when you're getting leaky blood vessels, shown up by fluorescein angiography. So as you can see, you get little bits of eye that have lit up. When you then look at this one, these are, unfortunately, they're not all at day 85, but they're close to the end of the study. And even with the eye of faith, you think there's less fluorescein lighting up here. So it looks like it does something. We sent this data to Belfast, got them to, in a masked fashion, analyze it for us, and the results were really good. If you compare our screening visit to day 8, and red is placebo and blue is our active drops, that you find there's not much difference at day 8. We didn't expect there to be much difference because of the mechanisms of action. But if you compare screening to day 85, that actually you can see that the people who are on placebo, about 50% of them got an increased level of leakage. Their blood vessels at the back of the eye continued to leak, whereas the ones who were given active, 60% of them got an improvement. So it's not that we're just holding the disease, we're actually stopping it in its tracks. This, again, I'm going to ask you for the eye of faith. This is where your macula is. These are big, blobby black bits of edema. And that is the beginning screening. This is day 8, not a lot of difference. Then you come on to day 29. Peripherally, it's beginning to get a little thinner, a little less edema. Come to day 57, it's getting a lot better. By the time you get to day 89, there is no edema. We were unable to measure visual improvement in these patients because they hadn't lost any, so there was nothing to improve. But we think that it is caused by edema in the eye, and therefore we think that it should improve in patients who've actually lost some. So, basic results: it reduces signs of non-proliferative diabetic retinopathy, i.e. leaky blood vessels, and the central macular thickness, which is the key measurement everybody's looking at, decreases. We've got a plan for our phase 2, which is what we want the money for. So the FDA, in whom we've had informal chats, have said we can do a 6-month study. So that's what we're going to do. We're going to increase the dose. The first study was done with 1.5. We're now going to do 3 and 5 milligrams and see if in patients who are more severe in their disease, if we can get a bigger drop in thickness of macula and we can see an improvement in vision. And then we will be moving on to phase 3. This study is designed to give a go, no go. It either works or it doesn't work, because we know it's safe, well tolerated. We think it works. If it doesn't work in this, if it doesn't improve vision, there is little point to continuing with this. We have to show people's vision improves. So we think we have a... Well done, that man. We think we have a very good product. We asked physicians working in ophthalmology here in Europe, but also in the States. We've got... That was mostly because VC said, nah, they'll never stop putting needles in people's eyes. That shows you the mentality of a VC. However, and I've been one, so I can say that, they said to us things like, I didn't do medicine to stick needles in people's eyes. I think you might have the Holy Grail. It was very positively received. These patients, early stage patients, are getting no treatment. So we're not kicking something into the sidelines that everybody is happy injecting. These patients are hanging around waiting to go blind, and we think we can treat them. You don't really need to see that one. This is just what we're going to spend the money on. suites.